Hey everyone, I'm Ron Alfa, founder and CEO of Noetik. So what we're building is frontier AI for human biology. I'm really grateful for Daphne for actually setting up a lot of the material I'm going to talk about today. Let's see, get that going. So everyone's here in San Francisco. I'm sure you're all believers. AI is really transforming every aspect of our lives. I know everyone's using LLMs. Probably half the people in this room took a Waymo here, and we're on the verge of an autonomous workforce. So we're there. But arguably, the next frontier that we're making much slower impact on is human health and biology. And so really to be able to train models that can help us to understand biology and disease, we need to be able to build models that understand human biology really from the level of the genome to the cell, all the way up to the tissue in patients. And that's what we're doing at Noetik. We're really trying to build models that can not only understand biology, but actually simulate biology so we can make better treatment decisions and actually develop better therapeutics. So the problem we're focused on is clinical translation. So translation means which drug works in which patient, essentially. This is the biggest problem in drug discovery today. If we can increase the probability of success for therapeutics in the clinic, we can make a huge impact for patients and radically reduce the cost of drug discovery. So this is kind of an important problem. It's also an important problem for us in terms of building these models. Because in order to solve the translation problem, we need to bring in the data that allows us to train models that understand human biology. So what we are doing is sourcing essentially clinical data sets that allow us to train models that are essentially perturbation systems using actual human clinical data. It all starts in our labs. Again, Daphne made this point really nicely earlier today. For most of biology, we just don't have the data. So when we founded the company, we said we're going to generate this data from scratch. So we are sourcing human tumor specimens, thousands of them, to our labs in South San Francisco. And we've developed really a custom workflow to process these tumor samples into a fit-for-purpose data set for machine learning. And so what makes us unique is we're generating many different layers of data. So from each tumor sample, we get the pathology layer, we get the cellular layer, and we get spatial molecular layers like transcriptomics and genomics. And so essentially we're able to give these models everything they need to learn biology from the raw data themselves. And so here's a simulation, or demo of one of our virtual cell simulator. And this is actually real data here. And what we've trained this model to do is simulate the biology of a cell in the context of a human tissue. And so what I'm showing you, on the right here is a patch of human tumor tissue. And what we're simulating is an immune cell. And what we're going to do is place the immune cell across, iteratively across every portion of this tumor cell. And the model's making a relatively simple inference. It's looking at the expression of any of thousands of genes as a function of the context of that immune cell, the location of the tumor, or the location in the tumor, the cells surrounding that particular cell, and the particular patient context. And what you can see from the heat map that the model generates is there's some very specific context-dependent information. So gene expression for this one gene that we're showing can vary depending on where this immune cell resides, or the virtual immune cell resides, in this tumor sample. And so there's really interesting information that's going to depend on a lot of the context of the tumor biology here. So this is really interesting in sort of a demo for a single patch of a single tumor for one patient. How do we build this up to really begin to make treatment decisions? Well, with a lot of GPUs, we can essentially scale this inference across billions of inferences, essentially across many different patients. Thousands of different genes, and then we can begin to get to what we think of as a representation space of tumor biology. And so again, this is real data here. These are from our lung cancer cohort. So roughly close to 2,000 patients worth of lung cancers. And what we're showing you are different representations of tumor biology that are described by the model or learned by the model in a self-supervised way. So we're not using any labels here. Really, the model's learning about underlying tumor biology from the raw data. And I won't get into the details here, but what's really remarkable about this is, these groups represent, biology that we know is interesting. This isn't, a black box. We have the virtual cell simulator. We can understand why the model is calling these different patient subtypes different. And we can actually look and see, is there any known biology that the model's finding that can help us sort of know that we're on the right track? And it turns out that the model nicely identifies a set of patients in the top there in gray that we know respond to current approved immune therapies. And again, this is not with the labels. The model is simply looking at the underlying biology and finding these patients that we know overlap with the patients that would respond to these immune therapies. So we could actually take this type of model and we could train a classifier and we can make better predictions about who's going to respond to these approved drugs in the clinic. But more interestingly, we can begin to look at some of these other patients and ask, are there therapies that are not yet approved that we can use to treat some of these other cancers? So the approved drug only treats about 12% of lung cancer patients. So if you look across all of these other patient distributions, there are many other patients with different biology that we really have no clue how to treat in the clinic. And so what we're focused on today is looking at clinical stage drugs that have been in some number of patients and where there's some activity, but we really haven't been able to solve that problem of who are the patients that are going to respond. And we're trying to map those clinical stage drugs to some of these patient populations that we're identifying. Now, I started this talk saying these are frontier AI models of biology, not necessarily like drug discovery. So I'll end with just a single slide here to emphasize that these capabilities really go beyond drug discovery capabilities and can begin to expand into other areas of medicine. So this is some work that we're doing on an ongoing basis that is focused on asking whether we can use this virtual cell model to interpret standard clinical pathology slides. So the image here shows a pathology slide. These cost pennies to generate. They're generated in millions of hospitals around the world for everything from cancer to many other diseases. And pathologists can look at these and they can discern essentially rough tissue architecture. And pathologists have gotten good at identifying diseases from that architecture. But you really don't get a depth of molecular or cellular information from these images. What we've been able to do is use these images as input and use our virtual cell model to essentially infer both cellular biology as well as gene expression from these pathology images. You can then get essentially a region description. So that's what I've got highlighted there, 3 different regions. We think of them as essentially concepts. The model is defining concepts of these tissues that are described by essentially gene expression signatures. And then we can then take those signatures and give them to an LLM. And then the LLM can produce essentially a descriptor of what's happening in that region of the tissue that goes far beyond what a pathologist would be able to do with just looking at tissue architecture. So essentially you turn this very inexpensive assay that costs pennies to generate into an assay where you can now generate insights that would cost you thousands of dollars to generate from a transcriptomics assay or others. So this type of application is incredibly powerful, and we could think about applying it to many areas beyond oncology across healthcare. So thank you very much. I'm Ron Alfa. We're Noetik, and really our goal here is to build models that can help us discover the right drugs for the right patients, and help us treat patients across all of medicine, beginning with oncology. Thank you.